Welcome to Science Era. In this video, we're going to focus on chapter number 7 of FIS 1601 module, the nervous system. Objective for today's video is organization of nervous system and nervous tissue. We're going to look at the structure and functions of the nervous tissue. Let's begin. Introduction to nervous system. Nervous system uses its million of sensory receptor to monitor changes occurring both inside and outside of the body. These changes are called stimuli and the gathered information is called the sensory input. It processes and interprets the sensory input and decides what should be done at each moment, a process called integration. It then causes a response or effect by activating muscles or glands, effector via the motor output. We're going to look at every step in detail. Organization of nervous system. Structural classification. The central nervous system consists of brain and spinal cord, which occupy the dorsal body cavity and act as the integrating and command center of the nervous system. They interpret incoming sensory information and issues instruction based on the experience and current condition. Dorsal mean at the back. The peripheral nervous system PNS includes all part of the nervous system outside the CNS. It consists mainly of nerves that extend from spinal cord and brain. Spinal nerves carry impulses to and from the spinal cord. Cranial nerve carry impulses to and from the brain. These nerves serve as communication line. They link all part of the body by carrying impulses from sensory receptor to CNS and from CNS to the appropriate gland and muscles. Before we move on, if you are new to Science Era, welcome. Every day, new videos on various subjects will be added with a special concentration on the UNISA module. Don't forget to hit the subscribe button at the bottom of the page. Let's continue with the organization of the nervous system. Now we're going to look at the functional classification. Sensory or efferent division, a nerve fiber that carries impulse to CNS from the sensory receptor located throughout the body. Somatic sensory fibers delivering impulses from the skin, skeletal muscles and joint, and visceral sensory fibers transmitting impulses from the visceral organs. Peripheral nervous system is divided into sensory, efferent division and motor or efferent division. Motor or efferent division. Nerve fibers that carry impulses from CNS to the effector organ ossicles and glands bringing about a motor response. Somatic nervous system, conscious control of the skeletal muscles, it's voluntary, and autonomic nervous system, ANS, regulates the activity that are automatic or involuntary. So, PNS is divided into two parts, sensory or efferent division, motor or efferent division. Sensory division includes sense organs. Motor division is further divided into somatic and autonomic nervous system. Somatic nervous system is voluntary while autonomic nervous system is involuntary. Autonomic nervous system is further divided into sympathetic and parasympathetic nervous system. Sympathetic nervous system is fight or flight. Function during extreme situations, for example, increased heart rate, rapid breathing, cold, sweaty skin, dilated pupil. They are all the functions of the sympathetic nervous system. Parasympathetic nervous system is when the body is in resting and digesting state. Most active when body at rest causing normal digestion, voiding feces and urine. Goal is to conserve energy. Nervous tissue structure and function. There are two types of cells: neuroglia. It is a supporting cell not able to conduct impulses, can undergo cell division. Most brain tumors are gliomas formed by the glial cells. Then we have neurons, nerve cells that transmit impulses, functional unit of the nervous system. Glial cells are there for support, insulation, and protection. 90% cell in brain are the glial cell. Central nervous system contain four types of tissues: astrocyte, microglia, ependymal cells, and oligodendrocyte cells. And peripheral nervous system, or PNS, contain two types of cell: Schwann cells and the satellite cells. Now we're going to look at the each type of cell separately. First one is astrocytes. Astrocytes are the star-shaped cells, account for over half of the neural tissue. Their numerous projections have swollen ends that cling to the neuron, as you can see in the diagram. Their function is to brace and anchor neurons to capillaries, intermediary cells between neurons and capillary. They aid in exchange, protects neuron from harmful substances present in blood, control chemical environment in brain by picking up excess iron and recapturing released neurotransmitters. Then we have microglia cells. Microglia cells are phagocytes that defend CNS cells. They are spider-like phagocytes dispose of debris or dead brain cells and bacteria. They are the smallest of glial cells. Then we have epidermal cell. Ependymal cells are lines the cavities of brain and spinal cord. Their function is beating of cilia help circulate the cerebrospinal fluid that fills cavities and form protective cushion around the central nervous system. The next one is oligodendrocyte cells. It wrap their flat extension around axon for many nerves, form myelin sheet, which is fatty insulating covering, protect and cushion nerves. It speeds up nerve transmission, give rise to white matter of brain, and fewer extension. They have fewer extensions than the astrocytes. Last one is a satellite cell and Schwann cells, which are in the PNS or peripheral nervous system. Schwann cells are cells of PNS that myelinate axons, while satellite cells is protective and cushioning cells of the PNS neurons. Next is anatomy of neuron. First thing we look while discussing the anatomy of neuron is the cell body. The neuron's metabolic center is the cell body. It has a big nucleolus in its transparent nucleus. Except for centrioles, the cytoplasm surrounding the nucleus contain all the normal organelles, which confirm the amitotic nature of most neurons. In the cell body, the rough ER, also known as Nissl bodies and neurofibrils, intermediate filaments that help maintain cell shape, are particularly plentiful. Then we have dendrites, axon, and axon hillock. Neuron processes that convey incoming messages or electrical signals towards the cell body are the dendrites. So, dendrites are around the cell body, whereas those that generate nerve impulse and typically conduct them away from the cell body are the axon. Each neuron has only one axon, which arises from a cone-like region of the cell body called the axon hillock. Next is axon terminals and neurotransmitter. Only a few axon produces collateral branches along their length, but all axon branches profusely at their terminal end result in hundreds of thousands of axon terminals. Hundreds of small vesicle or membrane sac holds chemicals called neurotransmitter at these terminals. Next is nerve anatomy. In this section, we're going to look at synapse and myelinated sheath or myelin sheath. Synapse and synaptic cleft. Each axon terminal is separated from the next neuron by a tiny gap called the synaptic cleft. Such a functional junction where an impulse is transmitted from neuron to another is called the synapse. Although there are closed neurons, never actually touch other neurons. Central nervous system. Oligodendrocytes form myelin sheath in central nervous system. They lack neurolemma. The function is to protect and cushion nerves, increase speed of nerve transmission. Oligodendrocytes are located in CNS. Peripheral nervous system. Schwann cells form the myelin sheath in peripheral nervous system. Covering of most long neurons formed by wrapping of Schwann cells, specialized supportive cells wrapped tightly around the axon. Neurolemma is the outermost part of the Schwann cell which aid in neuron regeneration. Node of Ranvier is the gap of myelin sheath between Schwann cells located at regular intervals. Action potential jump from one node to another at the faster rate. Then we have neuron classified by the function. Sensory or efferent neurons conduct impulse from the sensory receptor in the skin or viscera or muscles to the central nervous system. Most cell bodies are in ganglia in PNS. Here you can see the sensory neuron on the diagram. Motor neuron is efferent neuron transmit impulses from the CNS, brain or spinal cord to the effector in the body periphery. You can see on the diagram motor neuron. Then we have interneurons. Association neurons complete the communication pathway between sensory and motor neurons. Their cell body reside in the CNS. Four things to remember about a neuron or nervous system is small collection of cell bodies called ganglia are found in a few sites outside the CNS outside the CNS and in the PNS. Bundles of nerve fibers, neuron processes running through the CNS are called tract, whereas in PNS they are called the nerves. White matter consists of collection of myelinated fibers and the gray matter contain mostly unmyelinated fibers and the cell bodies. Next we're going to look at the classification of neuron by their structure. So the first one is multipolar neuron. They have several motor processes. Neurons are all associated with other neurons. Most common neuron type is the multipolar neuron. They have dendrites, cell body and axon. Next is bipolar neuron. Bipolar neuron have only two processes on the cell body, axon and dendrite. They are rare in adults, found in, for example, eyes and nose. Then we have unipolar neurons. One process on the cell body, single process is very short process divided into the two. Peripheral process is the distal process contains dendrites on the end and the central process is the proximal contains axon terminals. Axon both peripheral and central processes conduct impulse in both direction towards and away from the cell body. Sensory neurons are located in PNS ganglia and they are all unipolar. Nerve impulse. Let's start with electrical condition of a resting neuron's membrane. Resting membrane is polarized, which means that there are fewer positive ions sitting on the inner face of the neuron's plasma membrane than there are on its outer face. In the resting state, the external face of the membrane is slightly positive, its internal face is slightly negative. The chief extracellular ion is the sodium ion, whereas the chief intracellular ion is the potassium ion. The membrane is relatively impermeable to both ions. Number 2. Action potential initiation and generation. Stimulus initiate local depolarization. A stimulus changes the permeability of a local patch of the membrane and sodium ion diffuses rapidly into the cell. This changes the polarity of the membrane. The inside become more positive and the outside become more negative at that site. Next, depolarization and generation of an action potential. If the stimulus is strong enough, depolarization causes membrane polarity to be completely reversed and an action potential is initiated. Then propagation of the action potential. Depolarization of the first membrane patch caused the permeability change in adjacent membrane, and the event described in step number 2 are repeated, which is stimulus initiate local depolarization. Thus, the action potential propagates rapidly along the entire length of the membrane. Next, repolarization occur. Potassium ion diffuses out of the cell as the membrane permeability changes again, restoring the negative charge on the inside of the membrane and the positive charge on the outside of the surface. Repolarization occurs in the same direction as the depolarization. Initial ionic condition is restored. The ionic condition of the resting state are restored later by the activity of the sodium potassium pump. 3 sodium ion are ejected for every 2 potassium ion carried back into the cell. So the 6 steps in nerve impulse are: first one, resting membrane potential is polarized. Number 2, stimulus initiate local depolarization. Number 3, depolarization is followed by the generation of an action potential, propagation of the action potential, then repolarization, and then the initial ionic condition is restored. Next is the transmission of signal at the synapse. Number 1. When action potential reaches an axon terminal, the electrical change open the calcium channels. Number 2. Calcium ion in turn cause the tiny vesicles containing neurotransmitter to fuse with the axonal membrane, and then at number 3, pores like opening form releasing the neurotransmitter into the synaptic cleft. Number 4. The neurotransmitter molecule diffuses across the synaptic cleft and bind to receptor on the membrane of the next neurons. If enough neurotransmitter is released, the depolarization will occur, generating a graded potential and eventually a nerve impulse in receiving neuron beyond the synapse. Number 6. The electrical changes prompted by neurotransmitter binding are very brief because the neurotransmitter is quickly removed from the synaptic cleft, either by diffusing away or by the reuptake into the axon terminal or by enzymatic breakdown. Next, we're going to look at the reflex arc. Reflex arc is the neural pathways which involve both CNS and PNS. CNS is central nervous system and PNS is the peripheral nervous system. Reflexes are rapid, predictable, and involuntary responses to stimuli. Once reflex begin, it always goes in the same direction. You can't reverse it. There are two types of reflexes: somatic reflexes and autonomic reflexes. Somatic reflexes stimulate the skeletal muscles, for example, pull hand away from the hot stove, while autonomic reflexes regulate the activity of smooth muscles like heart and glands, for example, secretion of saliva, changes in the pupil size, regulates digestion, elimination, blood pressure, and sweating. 5 steps involving in the reflex arc is number one, a receptor which react to a stimulus. Number two is an effector, the muscle or gland eventually stimulated, and then sensory and motor neurons to connect the two, receptor and effector, the synapse or interneuron between the sensory and motor neurons represents the fifth element, the CNS integration center. So the five elements of reflex arc is receptor, sensory neuron, integration center, motor neuron, and then effector. This brings us to the end of today's video. In the next lecture, we're going to discuss central nervous system and peripheral nervous system in detail. Thank you for watching. Don't forget to subscribe for more.